Okay, let's take a look at one of these articles, these peer-reviewed articles, the primary sources that questions might come up in for protein modeling. So this one is the programmable editing of a target base in genomic DNA without double-stranded DNA cleavage. So first off, that title gives us a lot of information. Programmable editing means that this is going to be some type of editing of DNA that can actually be targeted. It's not going to be random. It's going to be able to be specific and programmed. And then we're looking at a target base. So it's going to be looking at one base, and one base means one nucleic acid, one nucleotide. And then it says in genomic DNA. So this is changing one specific base in DNA. And then also without double-stranded DNA cleavage. Now, normally CRISPR-Cas9 will make two cuts. Each cut will happen on one of the strands of double-stranded DNA. So this is editing DNA without cutting it. And normally before this, CRISPR always did a double cut of DNA. So let's look at this first area that I've highlighted and my little post-it note that goes with it. So it says current genome editing technologies introduce double-stranded DNA breaks at the target locus as a first step to gene correction. So that means that normally CRISPR would make a cut straight through both strands of the DNA molecule at a specific sequence. And that was the first step to correcting that gene, was to make the double cut. The next sentence, although most genetic disease arise from point mutations, current approaches to point mutation corrections are inefficient and typically induce an abundance of random insertions or deletions. Insertions and deletions are going to refer to now throughout this paper as indels. That means insertions or deletions at the target locus resulting from cellular response to double-stranded DNA breaks. So that means that although most genetic disease happens from one mutation to one nucleotide, their current approach of cutting that DNA against both strands and then after that cut happens, the cell tries to repair, and when the cell tries to repair that break, we get some insertions or deletions of nucleotides. And that is a very inefficient way to correct a point mutation, because it could just lead to other mutations as well. So, and then here's my note that is before programmable editing, random mutations would occur at sites where CRISPR-Cas9 caused a double-stranded break in the DNA molecule. These point mutations are often harmful and cause nucleotide insertions or deletions when the cell would attempt to repair the break. So that's the history of CRISPR-Cas9 before it became this targeted base editing tool. So the next portion here, we report the development of base editing, a new approach to genomic editing that enables the direct, irreversible conversion of one targeted DNA base into another in a programmable manner without requiring double-stranded DNA backbone cleavage or a donor template. So they're saying they have developed a new type of base editing that is programmable at a specific sequence. You don't need to have a donor template, and you don't require to double cut that DNA at all. So here's my next note. My next note, you can also see it on the right, because I noticed it's not all showing up in the screencast, but it says the enzyme cysteine deaminase was attached to CRISPR-Cas9. So that's, let me see if I can expand this a little bit. Okay, so how they changed CRISPR-Cas9. They added an additional enzyme to CRISPR-Cas9, and that enzyme's name is cysteine deaminase. So that resulted in the ability to have CRISPR-Cas9 target specific DNA sequences like it always has been, but now it no longer cuts the DNA molecule. Instead, the enzyme removes an amine group and that's where we get that part of the name deaminase. So the NH2 is an amine group, and it removes it from cysteine. Now, cytidine, that is the nitrogenous base of cytosine. So that's like the cysteine nucleic acid that we all know. So that's where the enzyme cytidine deaminase means it's going to remove the NH2 from cysteine. So once that happens, that cysteine molecule is now uracil. The uridine, that's a nitrogenous base of uracil. So what this enzyme actually does is it changes a C to a U in DNA. Now, we know U's are only found in RNA. They're not found in DNA. So the cell will normally want to repair that. So when repair enzymes from the cell remove the RNA nucleotide uracil, they replace it with thymine because the cell knows, hey, that's uracil. Uracil is only in RNA, it's not in DNA. Let's replace that with thymine. And then on the opposing strand of the DNA molecule, remember thymine's normally bonded to adenine. So if we change a C to a T, the other side of that molecule that it's hydrogen bonded to is going to change from guanine to adenine, from G to A. And then let me just go ahead and bring over these nucleotides so we can take a look at them. So what is happening is here's cytosine. So this is in DNA and RNA. And then let's look over here. This is uracil and RNA only. So notice like there's a lot of similarities here. Excuse me, but one thing that is different is this amine group right here, this NH2. So if that is removed and that enzyme removes that amine, it changes it here to uracil. And then when the cell identifies that there's uracil in its DNA, its repair enzymes switch out the uracil for thymine, which is right here. So the overall end result of that enzyme that's now been attached to CRISPR-Cas9 is that C is now changed to T with the removal of this amine group right here. So some other things with this. This is very efficient, and they're also saying that this could be a good technology to use to correct a variety of point mutations. A point mutation is one change in one nucleotide in DNA, and that could be relevant to lots of different human diseases. So moving down a little bit, let's look at the next thing that I highlighted. This portion right here, I just wanted to remind you that these sentences that are highlighted, we can model that looking at the HHMI website where we can it made that three-dimensional model of CRISPR-Cas9 and how it worked with the PAMs and all of that. So we know CRISPR has been widely used to mediate genomic editing in a variety of organisms and cell lines. CRISPR-Cas9 protein, its RNA complex localizes to a targeted DNA sequence through base pairing with a guide RNA, and it creates double-stranded breaks at the locus specified by the guide RNA. And this is how we've known that CRISPR has always worked, and that's what is in the HHMI interactive. So now we know that most genetic disease requires a correction of a point mutation, not just a random disruption to the gene. And that's what CRISPR-Cas9 had always done, is it just randomly disrupted the gene. So instead of correcting a mistake, it would just destroy that gene and it wouldn't work any longer at all. Okay, now as we continue to read here, okay, so still looking at this first page in the second column, the last thing that I've highlighted here is this information about the different enzymes that they investigated. So these few sentences say that they looked at 4 different cytosine deaminase enzymes. They looked at two different enzymes that humans have, one from a lamprey, which is a very like simple invertebrate, and then they looked at one from a rat, and what they ended up finding is that the best enzyme activity was found in the enzyme from the rat. So knowing that, they took that enzyme from the rat, that cytosine deaminase, and they attached it to CRISPR-Cas9 by attaching the amino terminus of the rat enzyme to the carboxyl end of the Cas9 protein. Because remember, when we put amino acids together, an amine group always attaches to a carboxyl group, and then we have a dehydration synthesis reaction occur. So they had that dehydration synthesis reaction between the N terminus of the CRISPR-Cas9 protein and then the carboxyl end, or the C terminus, of the rat enzyme. And then that's what they're showing here. Here is the Cas9 protein in blue, and then here is the rat enzyme, the cytosine deaminase, and it's connected right here to each other. And then what this is showing is that they have had this guide RNA that identifies the cytosine in a targeted area that is upstream of a PAM. And once that occurs, we do not cut any longer because the cutting ability of the Cas9 protein has been stopped. Instead, what this does is it changes the C to a U, and then when the DNA replicates or when it repairs, then we have the U changed to a T, and then the opposing side changes to an A. So we have successfully here edited this base. And then we have some electrophoresis results here that are looking that this is data that supports the model that they've designed for us here. And then over here, they're looking at the effects of different targeted C positions in base editing. So they're looking at, well, what if the C is by a G, or two C's together, a C and a T, an A and a T, like which one does the enzyme do well in? And then all of this here is talking about treating different DNA sequences to make those point mutations, and then it's seeing if those point mutations translate into different amino acids when a protein is created. Because we changed the DNA, which we assume will alter the messenger RNA, and then that will affect translation, which means that we will get a new amino acid instead. And what they have found with all of that data here is that these results show that the new base editor that they created is efficient, and it can precisely edit an amino acid. So they can change an amino acid in a protein even when the results in the DNA is only less than one nucleotide. So one nucleotide change in DNA can successfully change an amino acid in a protein, which we know. Sorry about that. And then what this is showing is that they're looking at p53 mutations. And one thing that we've learned about in AP Bio is that p53 protein is a cell cycle regulator, and that is a protein that acts to inhibit many cancers from forming because it stops the cell cycle from occurring. So if there's a mutation in p53, that's been linked with several different types of cancers. So p53 protein, which we know is a cell cycle regulator, it inhibits cancer. That was found even before DNA. Those changes were found to that protein before DNA was replicated or repaired from the U to a T. So the effects of the base editing in p53 have been very promising, and especially knowing that this p53 mutation is heavily associated with different cancers, that is, you know, a promising tool. And then finally, the last sentence here talks about like for future research, or where like things may go or what they might do in the future. So now that we know that an enzyme can be attached to CRISPR-Cas9 that can change a cytosine to a uracil by removing an amine group. They're saying, you know, maybe we can look at some methylases and demethylases. Those are enzymes that can either add, a methylase would add a methyl group, and a methyl group is a CH3, or a demethylase, which would remove that methyl group. And they're seeing like that could be another tool that could edit some of those DNA base pairs. But also here, because we could, like some nucleotides have methyl in them, but the last thing is important. It says and epigenomic base editing. So the epigenome, that is talking about what determines whether a gene is silent or if it is expressed. And we know many things control gene expression, but the epigenome is one of those. When a gene is silenced, it is condensed and it does not go through transcription. That happens when methyl groups are added to the DNA strand. So the methylation or adding of methyl groups acts to silence a gene. So if we could remove methyl groups, we could turn genes on. We could also add methyl groups to turn different genes off. So this is promising too. Like, and let's say an example of this. There are some people who are lactose intolerant, but they drink milk from their mothers or formulas just fine when they were babies. But now that gene that breaks down lactose isn't expressed any longer because it's been silenced, right? So there are some genes that we have that aren't working properly because they're silenced genes, because they have methyl groups attached. We could remove those methyl groups now. In the future, if like this is one line of research, or we could add methyl groups if we have a gene that we would want to silence, so it would no longer make a protein. So I hope this helps, and I will do the next scientific journal article soon.